The cell cycle will happen many times in the cell's life. This is the way cells reproduce. G1 is the first step of the interphase. It takes 5 hours and the cell grows in this step. The next step is synthesis, or S. It takes 8 hours and it duplicates the DNA. Condensed DNA will now form 4 pairs of chromosomes instead of just 2. We can observe in the image how the first cell has only 1 set of DNA and the second cell has 2 sets of DNA. The last stage in the interphase is G2. It takes 6 hours and it synthesizes proteins and duplicates organelles. In this image we can see the first cell with its 2 sets of DNA and the second cell with its 2 sets of DNA but with extra proteins and organelles. Mitosis takes 50 minutes. It is a step where the cell divides. This creates somatic cells only, which means that the cells will be exactly the same and they are not apt for reproduction. Mitosis begins with prophase, where the nucleus disappears, chromosomes are created, and these chromosomes pair with a sister chromatin. Then we have also the formation of the spindle, which are microtubules. In this image, we observe the nuclear membrane dissolving. We can also see the 4 axes of the DNA. These are paired sister chromatids. As we can see in the amplification, chromatin is the unraveled DNA. Then it condenses into a chromosome. Then this chromosome pairs with a sister chromatid and forms the X. We can see the spindles at the bottom. These are microtubules. Metaphase follows prophase. During the metaphase, the sister chromatids move to the middle of the cell, and spindles attach to the centromere. In the image, we can see how the DNA has moved to the center of the cell. The spindles have attached to the point where the sister chromatids are joined. This is called the centromere. Next comes the anaphase. During the anaphase, the centromere is split and the sister chromatids are pulled to opposite sides of the cell. In the image, we observe how the spindles have pulled the sister chromatids apart by breaking the centromere, and they are pulling them to their pole of the cell. During the telophase, the spindles break. A new nuclear membrane envelops the chromosomes, and the chromosomes unravel into chromatin once again. We can see in the image how the cell is about to split. The spindles are broken and no longer attached to the DNA. Around the DNA, we see a new nuclear membrane on both sides. We also see that the DNA inside the nucleus has unraveled from chromosomes into chromatin once again. The last step of mitosis is cytokinesis. This is simply when the cell divides. We can now observe the 2 cells, product of the single cell that underwent the cell cycle. Meiosis is the other way to divide cells. It takes 90 minutes and it is for sex cells only. It is a 2-part cell division and it produces 4 cells with 1/2 the genetic charge. We need 1/2 the genetic charge only so that the sex cell can combine with the genetic charge of the mate, and this way the baby can have a full charge with half of the mother and half of the father. Prophase 1 is basically the same as a regular prophase. We have some extra steps. In the leptotane stage, chromatin condenses into chromosomes. We also have this step in regular mitosis. In this image, we can see the chromatin condensing into the chromosome. The zygotane stage is also present in mitosis. This is when chromosomes attach to their sister chromatin. The next step, packetane, is essential for meiosis. In this step, the chromosomes cross over to blend the genes. By blending the genes, we obtain chromosomes with a very diverse charge. This way, the offspring can have the best genes of the person. In this image, we can see how the sister chromatids are together, and in the amplification, we can see how they are crossing over. That's what the arrows mean. In the stage diplotane, the chromosomes stop blending their genes, but they stay together. We can see in this image that the sister chromatids are still together, but they have stopped blending their genes. Diakinesis is the last stage of prophase 1, and this is when the sister chromatids separate from one another. At the end of prophase 1, we will have also dissolved the nuclear membrane and formed spindles from microtubules. The rest of meiosis 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1 are basically the same thing. Before starting the second stage of meiosis, meiosis 2, there is a phase of rest called endokinesis. After endokinesis, we can begin meiosis 2, which is formed by prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. These steps are also the same thing as in mitosis.